Chapter 4 takes a look at cells. So the last couple of chapters we looked at biochemical molecules, and now we're going to look at the basic component of all living things. When scientists define a living thing, one of the components or requirements for a living organisms, they must be made of cells. So when we look at cells, we look at the structural components of cells. All cells, have a basic cell membrane and other components inside, structural components that have very specific functions. Not all cells are shaped the same. Some cells are shaped due to their function. For example, this is a respiratory cell, nasal respiratory cell, and notice it has some structures that not all cells have, which is cilia that actually beat back and forth for the purpose of trapping small particles before they actually enter your lung tissue, because that is where you exchange oxygen. Here are some other one-celled organisms that actually have cilia. They are only one cell. They can live on their own. Here's another example of structure and function, the sperm cell compared to the large egg cell. So cells are us. Here's some more examples of different types of cells of the human. We have red blood cells, white blood cells, each serving a specific function. Red blood cells help deliver oxygen to all the cells of our body so we can make energy. And our white blood cells, of course, are our defense against pathogens. The nerve cell, its structure, it's a branching out structure from its cell body because it actually receives information in the form of neurotransmitters to help us to respond to our environment or stimuli. So how many cells do we actually contain? A person contains 100 trillion cells. That's a lot. There are about 200 different types of cells in mammals, one of us. And they're very tiny, measured on the average of approximately 20 micrometers across. That's about 1,250 cells, so shoulder to shoulder per inch. The cell theory was established and proposed by two main scientists in around 1838-1839. These two scientists are Schleiden and Swan, and the cell theory components are the following. All organisms are composed of one or more cells. Cells are the smallest living things. Cells arise only by division of previously existing cells, and all organisms living today are descendants of an ancestral cell. If we look at the scale and abundance of different types of cells, bacteria are very small. So if we look at the magnification from 85 times its size to 425 times the size, all the way up to magnified to 2,100 times its size, you can get an approximate idea of how small cells can be. There are fundamentally two different types of cells, prokaryotic and eukaryote. So when we look at this prokaryote, karyote means basically cell, pro means the beginning. So when we look at these are very simple cells that existed on the earth at the beginning of its time. These are the only living things on earth that are made of the cell or bacteria. So if you look inside this cell compared to this cell is called a eukaryote. The prefix EU stands for true. So what do we mean by a true cell? Well, a true cell, in simplistic form, means it has a true nucleus. So what is a true nucleus? Well, a true nucleus means that genetic material inside, that DNA, is separate from the other components of the cell, the cytoplasm. So this is a true cell because it actually has a nuclear membrane that separates the DNA from the rest of the cell. Compared to this cell, its DNA is just kind of floating around in the cytoplasm, okay, in contact with their little tiny structures inside called ribosomes. You also notice that there's plenty other structures in this eukaryotic cell. They are all separate components, separated from the cytoplasm by a membrane. So here are mitochondria, centrioles, endoplasmic reticulums, Golgi bodies, vesicles. All of these this is smooth ER, rough ER, Golgi bodies. And the main inside is a watery base that has a mixture of proteins in it that kind of holds the cell together, and it's surrounded by a cell membrane. Here we still have a cell membrane. So both of these are cells. They differ in that this one does not contain membrane-bound structures, whereas the eukaryote is a true cell that does contain membrane-bound structures. So when we look at some more differences between prokaryotic and eukaryotic cells, we notice a few things. We don't call the nuclear material in a prokaryotic cell a nucleus. It's actually called a nucleoid because it doesn't have a membrane around it. Whereas the DNA in a eukaryotic cell is membrane bound. Size. Prokaryotic cells are very much smaller than a eukaryotic cell. Organization. Single cell, often multicellular. Metabolism. Some bacteria or prokaryotic cells do not need oxygen. They can live in an anaerobic environment, whereas all eukaryotic cells usually need oxygen to exist. And then, of course, this big word, organelles. There are no membrane-bound structures called organelles in prokaryotic cells, whereas those membrane-bound organelles do exist in our eukaryotic cell. Here is an idealized animal cell. What do we mean by idealized? Well, when we look at this cell, this is a model. Remember that we have 200 different cells of our body. Therefore, each of them will be structurally different, and they also may contain or may not contain certain organelles, depending on what they do. So let's just take a look at this little model and look at some structures inside of this animal cell. So if we go from the inside toward the outside, we see a membrane, plasma membrane that controls what enters and leaves the cell. We see a watery base called the cytosol, along with a few little proteins within, making it like a gel-like substance, and that's the proteins. It's called the cytoskeleton. We also see other structures. For example, Golgi complex. These basically are areas where certain chemicals are made and sent there to be changed up a little bit, packaged up so that they can actually leave through this little vesicle and leave through the membrane because they're too large. We see our mitochondria, the site of cellular respiration, for us to make energy in the form of ATP. And again, we see some transport vesicles where proteins are leaving this rough ER where proteins are made on ribosomes and transports them for export or to go along in the cell. We see a little different ER or endoplasmic reticulum here that does not have little ribosomes on it. That's called smooth ER. And smooth ER's main function is to make lipids or fats, and again, vesicles will break off and distribute the molecules either to the Golgi complex itself or directly out of the cell. We do see some free ribosomes within the cytoplasm, and again, ribosomes are where we have protein synthesis occurring. We have a few other structures here inside. We have our big nucleus, and remember it has a nuclear membrane, and here's what we call nuclear pores, where we see certain things that can actually leave the nucleus. One of them is mRNA, which we'll talk about later the year. The nucleolus is the site of where RNA is produced, the little center of the nucleus. And now this squiggly looking stuff is where your DNA is located. When a cell is not dividing, it looks like this, and we call it chromatin, or the DNA. If the cell is about to go through division, it will condense into structures called chromosomes. One more we want to take a look at are lysosomes. That is basically the structure that contains digestive enzymes that will break down large molecules. So it's kind of like our little garbage can. All right, when we look at those main divisions, again, we have our center, our nucleus, all the organelles that are embedded in our cytosol, our liquidy portion, that basically it has some proteins scattered throughout our cytoskeleton, and then the outermost part, that our plasma membrane. Here's a picture that shows a liver cell, and so you can basically see some color enhancements to show certain organelles. For example, mitochondria you can see right here. ER you can see real well here. You can see vesicles filled with glycogen, which is one of the things, remember, we talked about last chapter that the liver does. It is a storage place for glycogen energy cells. And then, of course, your nucleus, and then your chromatin and nucleolus. So when we look at the cytoplasm, again, it is a kind of gel-like substance, jello kind of, that contains of water and proteins. So when we look at the cytoskeleton, here's a visual for you. The red is the cytoskeleton, and then we have ribosomes scattered throughout there. When we look at our main eukaryotic cells, animal cells versus plant cells, we look at some major differences that you need to make sure you know. So here are some similarities, if you see here. But then when we look back, we want to make sure you understand that, remember, plants do not have, if multicellular animals, you know, we have bones or exoskeletons, whereas plants don't have these structural supports. So what they basically get their support from is they have something that animal cells do not, and that is a cell wall. And a cell wall is made of cellulose, a tough carbohydrate, which gives it support. Another thing you see inside are chloroplasts. Chloroplasts are green structures that contain chlorophyll, the site of photosynthesis, where this plant cell will make its own glucose in water, and actually can store that in a large vesicle that it can use the glucose in later. Because remember, plants do not eat, so they are autotrophs. So if we look and make some analogies here, we can look at, for example, a production facility. We can make an analogy that your nucleus is the control center, that the ER is a assembly line, it's making proteins. The workbench is where the site where those proteins are made, ribosomes. The distribution center is the Golgi complex, the cleaning crew, the lysosomes, the security gate, the cell membrane, the powerhouse, the mitochondria, and the structure itself, the cytoskeleton. The evolution of the eukaryotic cell. This is a theory of how eukaryotic cells came about, called the endosymbiotic hypothesis. It states that aerobic bacteria were once taken up by large cells, larger prokaryotic cells, and then became the mitochondria, which exists within the modern day animal cell and plant cells. Cyanobacteria were taken up then by other larger bacteria, and then became chloroplasts, which is the modern day plant cells. So if we look inside the nucleus, again, we see the nucleolus, we see nuclear envelope or nuclear membrane, and DNA. So when we look at this, remember that that little nuclear pore, DNA cannot fit through it, but it can fit a smaller type of nucleic acid called mRNA, which fits through there and is going to be involved in protein synthesis. So think of your nucleus as your control center. Alright, ribosomes and ER. So if you look in the structure here, ER, or endoplasmic reticulum, this has some little pathways or hallways, and this is basically the purpose of ER is to transport things. So when we look at rough ER, we call it rough because it contains ribosomes on the outside, and those ribosomes will be synthesizing or making proteins. So once the ribosome, here's a picture, makes these proteins, it will then place them into the ER to be transported and throughout the cell through a vesicle. Smooth ER is similar, except it doesn't contain ribosomes, and its main function is to synthesize lipids, and they transport again throughout the cell by vesicles and enter to Golgi bodies to be processed and changed up before they leave the cell. So, for example, inside the testes, these would be an example of smooth R producing the lipid steroid testosterone that needs to basically leave the cell through a vesicle. Golgi apparatus, think of these as your packaging facility. They contain stacks of saccules, and its main function is to take those simple proteins or lipids, change them up a little bit, add a sugar here, there, etc., and finally they will be removed from the cell to the outside of that cell. So one example of a vesicle that we're going to take a look at is a lysosome, which is involved, has digestive enzymes inside of proteins that is going to be necessary, kind of like your trash can removing old cell parts, or in a white blood cell it actually will break down a bacteria. So here are some kind of associations with organelle malfunctions. So one example would be cystic fibrosis. It has a genetic mutation that is going to be a malfunction in a protein itself in the cell membrane, and we will kind of talk about this a little bit later on when we do look at DNA a little bit more and how some mutations affect the production of certain proteins in the body. So the lysosome again is our digestive plant or trash can. It gets rid of worn out molecules, breaks down bacteria in white blood cells. So it is going to break in our waste products up and expel them from the cell. So here is an example of a white blood cell, this big thing here, and here's a little bacteria. So this cell membrane kind of forms what they call a pseudopod or false foot that will surround the bacteria, enter inside where the lysosomes will actually break apart that bacteria. Here's some examples of why do we study these little organelles, because if there's some malfunctions, we do see some, effects to the humans for examples of lysosomal storage diseases. Okay, the mitochondria powerhouse. This is, we'll take a look at it in another whole chapter when we study about cellular respiration. Notice that this is where, of course, we intake glucose with oxygen, change it up through chemical reactions, and we give off water, carbon dioxide, and we make ATP. So again, there's lots of diseases that cause muscle weaknesses and neurological disorders due to the malfunctioning of our mitochondria. Right, make sure we review the main differences between plants and animal cells. Of course, chloroplast is a big difference because plants are autotrophs. They were able to trap sunlight and use carbon dioxide and water and make glucose and give off oxygen so that they can actually use that glucose for their own cellular respiration purposes. Remember again that it has a cell wall needed for support made of that tough cellulose material, and this is the vacuole where the glucose and water and waste will be stored. So here is cells in the leaf. So if we look at this under the microscope, here are our chloroplasts. And again, another whole chapter will spend on looking at, the whole process of photosynthesis within this chloroplast molecule itself. So don't forget the differences: cell wall, central vacuole, and of course um, our chloroplast. So let's take a look for a minute at the vacuole. It not only stores water and waste, but it actually has to do with, what we call giving it support. So when we look at this vacuole itself, this is a nice, healthy plant, which says nice and turgid, but this one is flaccid. So what does that mean? Basically, it means that when that vacuole is nice and full of water, it puts pressure on the cell membrane against the cell wall and gives the plant support. Whereas if the vacuole is low on water, and not storing very much, the cell membrane shrinks inside a little bit, so the plant shrinks up and is what we call flaccid. All right, the cytoskeleton is basically made of many different types of proteins that gives the inside of the cell a gel-like consistency. There are three components. We have active filaments that interact with certain proteins and they can actually move. We have intermediate filaments that give it support, and we have microtubules which maintain shape and act as tracks along which organelles can move. An animal cell also has centrosomes, which is a pair of centrioles that's going to be active during cell division. So here are just a few views for you to kind of look at all the different proteins you see that exist within the cytoplasm or the cytoskeleton itself. So here's a cytoskeleton in action. It's kind of moving and gel-like consistency, taking up that bacterium. So here are a few others. Of course, cilia, and we see the flagella on the tail of the sperm. These are both specialized structures that basically move and will be for the purpose of action or movement. One tidbit here: smoker's cough in the respiratory cells is due to the destruction of cilia linking the airways. So again, cilia and flagella, are hair-like projections from the plasma membrane. They both act as are used for movement.